So tonight's video is going to focus on gene expression and the regulation of it, mainly the control of transcription, but we'll look at some other ways that gene expression can be regulated as well. And we're going to look at both eukaryotes and prokaryotes because it'll be a little bit different. So we'll be introducing some new vocabulary throughout this particular chunk of the unit. So the first thing we're going to talk about here is regulatory sequences. So a regulatory sequence is what its name suggests. It's a chunk of DNA that's going to interact with regulatory proteins to help control transcription. So for example, you can see in this picture here, there is a gene here, the yellow, that's encoding for the regulatory protein. So that then will produce this regulatory protein. And that regulatory protein then affects the target gene, whether that target gene is produced or not. So that's what it's referring to when it's talking about regulatory sequences. These chunks of DNA that basically control the regulatory proteins. And those proteins will then, in turn, control transcription for another target gene. In prokaryotes, gene regulation can happen very, very rapidly. Genes can be turned on and off very quickly. The benefit to that is it gives them some flexibility. It gives them some reversibility. So, for instance, if something was turned on, it can very quickly be turned off. If something was off, it can be very quickly turned on, which is important for that unicellular regulation there. And it allows them as well to adjust levels of enzymes for synthesis and digestion. Again, you have to remember prokaryotes are referring to a unicellular organism. So the one cell is in charge of everything. And so we'll look closer at prokaryotes at the end. We're going to focus more here in the beginning of eukaryotes. But again, some of your benefits of the way that prokaryotes regulate is that it allows them to turn things on and off quickly, and it helps adjust enzyme levels for them. In eukaryotes, remember we're referring to, when we're talking about eukaryotes here, as a general, we're talking about multicellular eukaryotes. And what we need to keep in mind with multicellular eukaryotes is I'm not trying to regulate just one particular cell here. I have to regulate the body as a whole. I have to coordinate the individual cells into the body as a whole, and as opposed to catering to just the needs of the particular cell. So gene regulation in eukaryotes has evolved to help maintain homeostasis. Remember that constant internal environment? So to help maintain homeostasis while the external environment is constantly changing. So that's kind of the reason why gene regulation has really developed in eukaryotes, is to help maintain homeostasis. Okay? And in addition, like I said, they're helping regulate the body as a whole. So we're talking about growth and development or things that are long-term. Okay? Gene regulation helps regulate those things. Okay? Specialization. So allowing cells to specialize. And by allowing a cell to specialize, that means we are turning on and turning off large numbers of genes. And by doing that, by turning on and off large numbers of genes, my eraser function isn't working, okay, that's going to allow the cell to specialize into a bone cell, into a brain cell, into whatever it needs to specialize, which will help coordinate, again, the body as a whole instead of just focusing on the individual cells. So gene expression can be controlled at many points. And again, here we are referring to a eukaryote. And I'm not asking, right now is not the time for you to memorize how all of this is going to work. Right now is the time for you to get this concept down of the fact that this pathway can be regulated, controlled, manipulated pretty much anywhere along it. And we're going to talk about these a little bit more in detail, some more than others. Okay? But right now, again, it's understanding this concept that anywhere within the pathway, this can be regulated or this can be controlled. Anywhere along the way from the gene, so from the actual DNA, to a functional working protein. So gene expression could be controlled during the packing or the unpacking of the DNA. Okay, that's going to be, you're going to see that happening up here. Okay? Basically, when the DNA is being uncoiled or it's being tightened, and again, we're going to talk about this first in just a minute. Okay? But the way the DNA is packed in there will dictate what can be transcribed and what is accessible for transcription and what isn't. Okay? So transcription is another spot where gene expression could be regulated. Okay? Also with the mRNA processing. Okay? If you remember, for mRNA processing, we did a lot of splicing, introns and exons, and removing those introns and splicing those exons together. Okay? So that could help control gene expression. Same with RNA transport. Remember, the messenger RNA had to be modified. It gets the 5' cap, the poly A tail, okay, and then it has to be transported out into the cytoplasm. So during that transport, okay, things could happen that could regulate whether that gene is expressed or not. Okay. During the actual process of translation. So during the actual process of translation, regulation could be occurring, as well as the protein processing. So kind of the final finishing touches of making it a functional protein, and then the degradation of the protein. How fast is that protein being degraded? Is it even if it's being made, is it being degraded so fast that it's not being used? Okay? Is it sticking around for a long time? Okay? But again, these are just all examples of places where gene expression can be regulated. So one of the first places that we mentioned that DNA could be regulated was with its packing and unpacking. So let's take a closer look at what that means, since that's going to be something that's new to y'all. We haven't talked about this before in the past. So let's kind of look. These here are just kind of our stair steps of basic DNA coiling and folding, not basic DNA structure made of a nucleotide, that kind of thing, but actually fitting all the DNA into your cell. Let's use humans as an example. If you took the DNA out of one of your cells, it is almost 6 feet long. It's about 2 meters long, so about 6 feet long, which is a huge, huge amount of DNA to cram into a little tiny cell. And so this is going to be basically how your cells are going to do it, how they're going to fit all that DNA in there. So the first step is the DNA forms that double helix. And we're familiar with the double helix, the 2 strands held together in the middle by the hydrogen bond. Okay, so we're usually familiar with that and okay with that. So the next step is the DNA will start wrapping itself around what we call histones. So histones are these little beads, so the little blue beads that you see in there, those are the histones. And the DNA double helix will start wrapping around it. You can see how, if you look closely, see that blue turquoise thread that represents the DNA? It's wrapping around those purplish histones. And so when the DNA wraps around the histone, it becomes a nucleosome. So these hunks right here are nucleosomes. So now I've started coiling the DNA around the histone. So I'm making it appear a little bit shorter by coiling it around the histone. And if you think about it, DNA that was wrapped around this histone bead would be more difficult to access to do transcription. DNA that's not wrapped around that would be easier to access to be able to do transcription. So you can see how this packing and unpacking can impact which genes can be transcribed at any given time. So once the DNA is wrapped around those histones to form those nucleosomes, now the DNA, those nucleosomes come together to form the chromatin fiber. If you think back to the cell cycle in mitosis, and we talk about during prophase, the chromatin condenses into chromosomes. So this is the looser form, the looser thread-like form. And you can see on the right here, these are all microscope pictures of what's happening. And so you can see this chromatin here is our looser form of the DNA. So these nucleosomes, though, you'll see are packed tighter together here in these chromatin fibers. And then these chromatin fibers will start looping on one another. So you can see it's forming, there's a protein base, and then you see it's forming all those loops, right? Just like villi do in the intestines to help increase surface area. And so now, in a very short space right here, I have been able to pack in a huge amount of DNA by forming those loops. So we'll keep building on each other. The double helix wrapped around the histone, which formed the nucleosome. Those nucleosomes wrap together to form the chromatin fibers. The chromatin fibers start bending to form these loop domains. And then those looped domains start getting folded together to form the chromosome. And you'll see this is the metaphase chromosome. That condensed chromosome, all that DNA is crammed into that one little chromosome. About 6 feet of it crammed in there. So looking a little bit closer at those histones, we're going to look at how we can wrap around it versus encourage it not to wrap around it. So again, the histone is that protein bead that the DNA is wrapping around to help form the nucleosome. This is in eukaryotic cells. Okay? Those histones have positively charged amino acids in them. If you think back to DNA from your freshman year when we did gel electrophoresis, and we talked about how DNA was negatively charged, and that's part of why it moves through the gel, is it's attracted to that positive charge and it's being repelled from the negative charge. Okay? And so if I have a whole bunch of positively charged amino acids within these histone proteins, then the DNA is going to be attracted to those, which will help it bind nice and tight and help wrap around those histones. So those histones with their positively charged amino acids attract that negatively charged DNA, and so they'll bind very tightly together. And how much they pack together, again, affects transcription, and we talked about this on the last slide. Very tightly wrapped around the histone, then I won't get any transcription happening and the genes will be turned off. Not wrapped super tight around the histone, then I can get some transcription happening. So we have what's called methylation. And so methylation will usually cause genes to turn off, meaning that there is no transcription happening with methylation. So for methylation, you can see it in the picture over here on the right. So with methylation, your cell has added methyl groups, you can see the little M's here, methyl groups to the DNA. And by adding those methyl groups to the DNA, that causes them to wrap tighter around the histone, which will turn the gene off and make no transcription happening. You can see how tightly packed in here those histones are. Where acetylation will do the opposite. So acetylation will usually turn genes on, because when I add these acetyl groups to the DNA here, that will actually cause it to loosen up around the histone. And so that will enable transcription to happen a little bit easier, and those genes can be turned on. Okay, so the way the genes are wrapped around the histone, methylation, adding methyl groups will wrap them tighter, therefore turn them off, where acetylation will add acetyl groups and it will actually loosen them, and so genes can be turned on. Okay, let's talk a little bit about epigenetic changes. This is one we're definitely going to talk some about in class, and we'll hopefully look at some articles. This is a very, very current field of research, constantly changing because it's happening right now. We are just learning these things. We are just discovering these things. This is one of those topics that when I was in high school, nobody even heard of this. When I was in college, nobody heard of this, and nobody was talking about this. And so this is one of those things that we don't have all the answers to, because research is coming out about it now. Okay, so epigenetics is just going to be our study of our heritable change in gene expression. So the gene expression the ability to express genes that we inherit from our parents, but that's not permanently changing the DNA. That's what it means by epigenetics. Remember, epi is upon or above. So these are not permanent changes to the DNA. These are usually caused by environmental changes, by social things that have happened to us. Okay? These can impact our DNA, and they can impact which genes are being expressed, but they're not causing permanent change to the DNA. So epigenetic changes, they can affect gene expression, and again, these can be reversible through reversible modifications of the DNA or of the histone. Again, this is not a permanent mutation DNA change here. So methylation and acetylation are both examples of epigenetic changes. If there is something in the environment that is causing us to pack on methyl groups, then we're not going to transcribe certain genes. If there's something in the environment that's having us pack on acetyl groups, then we could be transcribing genes, maybe genes that we don't want to, or maybe genes that we do want to. Okay? So these things can impact us, but they can also be passed on. They can be inherited, and so they can be passed on. So even if you pass them on, your children could potentially reverse them, or they could change them. And again, they don't always have to be bad. Okay? And again, I cannot emphasize this enough. This is not a mutation. This is something that can be reversed. It's something that can be changed. It is not a permanent change to the group or to the chunk of DNA. So these differences in gene expression can lead to cell differentiation. So cell differentiation is just going to be our process by which our cell attains its determined fate. So what is that cell supposed to be? Is it supposed to be a blood cell? Is it supposed to be a bone cell? So cell differentiation is when it reaches that state. Did it finally become a blood cell? Did it finally become a bone cell? And it's considered achieved when these observable traits can be seen. When we actually see the compact bone being laid down in the bone cell, when we see the adipose tissue and the cell arranged so that it can store the extra fat, when we see the modifications on a nerve cell, that's when these observable traits are being expressed. They're being expressed when genes are coding for tissue-specific proteins and when these observable traits are actually seen. The first signs that you're going to have differentiation occur is when we get the appearance of messenger RNA for the tissue-specific proteins. Remember, tissues are groups of cells that are working together, and so these tissues are going to be differentiated as well. And so when we first start to see messenger RNAs for these tissue-specific proteins, that tells us that the process of differentiation is occurring. So now we've got different genes that are turned on in these different cells. Different genes turned on, different genes turned off. Remember, every multicellular organism has the exact same DNA in every cell. I cannot stress that enough. Every cell equals the exact same DNA. And it's all about what's turned on and what's turned off that allows for this multicellular cell differentiation, cell specialization. So in a fully differentiated cell, transcription is my primary regulatory point for maintaining appropriate gene expression. So what that means is that in a cell that's fully differentiated, our primary point that we regulate things is what's being transcribed and what is not. There is no point in transcribing a whole bunch of genes to then process that messenger RNA to turn it all off. That's a waste of resources. So in these cells that have gone all the way through differentiation, that is going to be our primary regulatory point, is what are we transcribing and what are we not. So transcription factors are going to be playing a major role here with gene expression. And so as these transcription factors are introduced during development, this is going to cause sequential gene expression, meaning I turn on this gene, then this one, then this one, then this one, then this one. That is what's going to help these cells specialize and differentiate. So in eukaryotes, we're going to have groups of genes that can be influenced by the same transcription factors. And that's going to be important because it's going to help us coordinate and regulate the gene expression there. So it's going to allow us to, if I've got groups of genes that are affected by the same transcription factors, that allows for one transcription factor to turn everything on that will make it be a bone cell, turn everything on that will make it be a blood cell. Okay? So these transcription factors are going to help control, again, what's being transcribed and what's being not. So let's look a little closer at some of these. So some regions of control on the DNA are first going to consist of the promoter. So the promoter is going to be nearby the target gene. So if we look at this picture up on the right, you'll see our target gene is this blue box right here, and our promoter is right next to it. So it's going to be nearby. It's a nearby control sequence. So this promoter gene is controlling the gene. It's where the RNA polymerase binds and the transcription factors bind. If you remember, we have to have RNA polymerase to lengthen that messenger RNA. So this is where RNA polymerase can attach into the DNA, so then it can then move down the gene and do transcription. Okay, this will help determine our base rate of transcription, meaning that the rate that the gene is transcribed has kind of a set rate, and then that rate can be affected by other things that are impacting the gene. So there's our promoter region, controlling the DNA right next to it, allowing the RNA polymerase and the transcription factors to bind. You're also going to see these enhancer DNA sequences. So these enhancers are far away. So these are distant control sequences. Okay, so for instance, if I look back at this picture up here, I've got my enhancer far away from my target gene here. So it's a distant control sequence, and it will help bind to activator proteins. And these enhancers can help speed up the rate of transcription. You'll see in this picture here how this enhancer is folding back to come bind by the promoter and the target gene. So they do exactly what it says. They enhance the rate of transcription. Activator proteins will bind to this enhancer sequence, and they'll stimulate transcription. So you can see in the one in the yellow here on the left, okay, so here's my enhancer far away from here's my promoter right there. Okay, my target gene is right here. Okay, my promoter is folding back onto my ta-ta box there. And then my activator protein, this like circular mustardy yellow one there in the middle there, that activator protein is binding to the enhancer sequence and causing transcription to occur, where a silencer protein is going to do the opposite. So a silencer protein is going to bind to the enhancer and it's going to stop transcription. So this silencer, the pink one in the middle there, is preventing transcription from occurring. So their name will kind of tell you what they're going to do. But again, these are going to impact those enhancer sequences, and they're either going to activate transcription or they're going to silence and stop it. So eukaryotes, as we just saw, have those things that help coordinate and regulate gene expression between the promoters, the enhancers, the activators, the silencer proteins. Prokaryotes have those as well. So in prokaryotes, they are called operons. So operons are found in prokaryotes only, okay, and they are helping transcribe a single messenger RNA molecule. Okay, so they're transcribed in a single messenger RNA molecule only, and they have 3 parts. So each operon has a promoter region. This again is where the RNA polymerase binds, so the same as it was in eukaryotic cells. So there's my promoter region. It has an operator region. The operator region is right next to the promoter region. That's going to be your on-off switch. Okay. It's between the promoter region and the first gene. A lot of times it's multiple genes that are part of the operon. So it's between the promoter region and the first gene, and it will turn the whole thing on and off. Okay. And then the last part is going to be the genes that it controls. So you can see in this example here, there are 4 genes that it is controlling there. And so those genes, you'll notice I'm producing 4 different proteins here because it's all 4 genes are controlled. So in prokaryotes or in bacteria, the genes for the enzymes of a pathway will be clustered together. So they can be transcribed as just one long messenger RNA. So you'll get all the enzymes you need for the pathway based off of one transcription and translation situation. So it's beneficial resource-wise for the unicellular organism, for the prokaryote to do this. So for example, all these genes A, B, C, and D here would be all for the same pathway, so they can all make their proteins at one time together. Operons can also be what's called inducible or repressible, meaning that operons can be turned on or they can be turned off. Okay? They're either off and they can be turned on, or they're running all the time and they're on, and sometimes they need to be turned off or repressed. So we'll look at 2 quick examples of each one of those. So our first example here is what's called a lac operon. This is probably like the most common operon example, definitely the most common inducible operon example, but again, kind of your absolute 100% most common operon example in general. So a lac operon is an inducible operon, meaning that it is normally off, okay, and it can be turned on. It can be induced to be turned on. So if you look at the picture on the left here, you can see your promoter with your operator region right here, and you'll see that there's a repressor bound to that operator region. So this RNA polymerase gets stuck. So it don't make any messenger RNA because that RNA polymerase can't move down and do transcription. So in the absence of lactose, so when there is no lactose sugar available, this operon is turned off. I don't need to make lactase if I'm not consuming any lactose, right? That's a waste of my resources to be making these enzymes if I don't need to break this substance down. So if no lactose is present, this operon is going to be turned off, and the active repressor is bound there to the operator, stopping transcription. However, when lactose is present, or any isomer, so any shape that is any form of lactose, basically. These are details you don't really need to know. So when lactose is present, okay, it will bind to the receptors. You can see this little green one here. Okay, so this little green one, that's my lactose or my allolactose. It will bind to the repressor, and you'll notice it changes the shape of the repressor. So now that the repressor is kind of angled out to the sides, it won't fit in that operator anymore. And so it will not be turned on. It won't be turned off anymore. It won't be blocking the way. So now that RNA polymerase can travel all the way down. And so it can make all of the various proteins. It can transcribe all of the genes that are needed for the pathway to break lactose down. So that would be an example of an inducible operon. An operon that's normally off, that gets turned on, okay, in the presence of lactose, because now I need those particular enzymes that are used to help break it down. So the opposite will happen in what we call a repressible operon. So our most common example of a repressible operon is called the trip operon. And so this is an operon that is normally on. So you can see in the picture down here at the bottom, there's no repressor protein. The repressor is inactive, pointing kind of out to the sides, so it wouldn't fit in that operator, so RNA polymerase can go ahead and do transcription. And so this operon is normally on. The prokaryote is normally always making the enzymes that are needed to help make and produce tryptophan, hence the trip operon. However, if tryptophan is present, again, I don't need to waste resources on making all of these proteins. So it could get turned off. And so that's what you see happening here in the upper right. In the upper right, when tryptophan is present, it binds to the repressor, and you'll see it actually activates it. Different, opposite of the operon now. So now instead of it being kind of out to the sides where it won't fit, now it's straight up and down. The repressor can bind to the operator, and that will block the RNA polymerase. So I've repressed this operon, and then as tryptophan is consumed or disappears, okay, that will then release, okay, and that operon can get turned back on. So again, reversible and can happen very quickly and allow the prokaryote to do very quick changes and very quick regulation. Okay, so I know a lot of this stuff was new and not stuff you've seen before. So we will spend a little bit more time in class on these concepts, not necessarily just applying them, but also really kind of making sure we understand what an operon is, how eukaryotes can regulate gene expression with methylation, acetylation, that kind of thing.